Thank you. I'd like to thank the organizers for inviting me. I'm delighted to see such a level. I am not researcher, I'm a practitioner in functional medicine, integrative medicine, and I'm here maybe to contribute with my initial approach. Maybe it's not so scientific, but I would like to thank Dr. Sanchez for her work, and I'm going to represent certain aspects here in my intervention. I see that Spain is leading regarding the research on deamine oxidase, and maybe in France we're not in such a leading position. So today we're going to see the regulation of this enzyme, and I'm going to tell you as well that in my practice I analyze microbiota with a laboratory, and this has totally transformed my medical practice. So for those who are listening to us today, maybe they should take the initiative to develop in their current practice the dosing of these markers. So it's true that this is a topic that I am not an expert in or a scientist, but I know that today there are certain people and people who are not really aware of the activity of DAO. So I have some slides, of course, for the general public. So DAO, deamine oxidase, this is the enzyme that is responsible for the degradation of histamine. Histamine is produced from histidine, and as the professor was saying, and I'm also interested in the methylation pathway to produce histamine. I work with homocysteine with my patients that I dose systematically, especially when there's a histamine intolerance. And we know that this methylation is sometimes altered or disrupted. And we know that this is important because this HMT, this enzyme for methylation that is going to deaminize histamine, is very important. And we see that this is important at the liver level. And it's a cofactor for the degradation of this histamine. Now, another pathway that is also very interesting is the DAO pathway. So this is the digestive pathway because this DAO is present mostly in the intestine. We will see that there are other tissues, but it's mainly the intestine. And also there's another pathway. It's this histidine decarboxylase. And this can also favor this potential of histamine, this highly elevated potential of histamine. So in this case, there could be a DAO that is normal, but there can be this high fecal histamine. So we know that these are the modulation pathways for histamine. And of course, all of this is also related with these enzymatic cofactors. And the HNMT is an important methylation pathway. So I'm going to move quite quickly here because all this has already been presented. I've also been interested between the relationship between histamine and DAO activity, and there can be some variations and fluctuations, but I see that 1 out of 2 patients suffering from irritable bowel syndrome shows reduced activity of DAO. And maybe then we can discuss about the thresholds of DAO because, well, we do not seem to agree how to measure this activity of DAO, and certain labs establish that 10 is a threshold, and some other labs establish other values, and there's also the importance of the genetics in all this. And well, you know, in the case of genetic alteration, well, should we provide DAO supplementation? So actually, I do not have an answer, but I have seen patients who have DAO deficiency of a genetic origin, and depending on the lab which performs the analysis, and actually there's a lab that is partnering in this Congress, and fortunately enough, I could find this lab. I could find it because at the beginning, well, for 20 years, really, there were very few means to boost this DAO enzyme. And having a vegetable DAO is not very easy because, well, you know, patients are not attracted by animal origin enzymes. And here we can see that it's a study of our German colleagues, Schink, for instance, who have talked about a group of people who showed food intolerances and hypersensitivity syndromes and histamine intolerances. And we can see that these groups of population are quite similar in terms of their lifestyle and in terms of their symptomatology. They all have a leaky gut, and this is almost systematic. They have a dysbiosis and inflammation. And I thought, well, we should treat this histamine intolerance such as we treat those patients who are intolerant to gluten or to casein or other foods. And well, this somehow works, but it's true that this idiopathic histamine intolerance can lead to a difficult way of life because histamine is everywhere in all kind of foods. So it is quite a nightmare to try to find how to lead a low histamine diet. I have this patients who are intolerant to histamine, intolerant to gluten, and intolerant to some other foods. Well, it's very difficult to provide them with a balanced diet. So in this case, I would resort to vegetable origin DAO from a lab here in Barcelona because this will provide these patients with higher freedom in terms of their diet. But there are also some histaminogenic foods like chocolate and tomato and so on. Now, I also dose homocysteine and B2, B6, B12 vitamin because I think that this is a complete package. And when you treat patients who are histamine intolerant, we have to consider all the pathways, the activation pathways and the degradation pathways, because it's both that I produce too much histamine or maybe I have too much histamine because my DAO is deficient, or because I'm not methylating histamine properly. So I think that it's actually both pathways. And this is what our consultation tells us. And we also need to dose and to be able to measure the activity of DAO, and finding the right lab is not always easy. I have this lab with whom I work, and they measure the fecal DAO and the plasmatic DAO, and this is interesting as well. As I said, DAO is essentially present in the intestinal mucosa and also in the kidney. But it's not only histamine which uses the activity of DAO. There are other amines, and actually, for instance, I'm talking here about putrescine, cadaverine, and if you are intaking these other amines, there can be a very low DAO activity. And I also try to find binary metabolite. I do a lot of research in this field, and this is a marker for dysbiosis. And regarding the elimination of this biogen amines, this allows me to orient the treatment. For instance, there are some foods that produce putrescine, cadaverine. So here we could reduce some sort of animal protein in order to reduce this kind of amines. Mrs. Sonia Sanchez was already talking about these adverse effects of DAO deficiency, but yes, there's this diversity of symptoms that leads to a delayed diagnosis. And here in France, 80% or 90% of allergologists and gastroenterologists, which keeps saying that, you may have brain fog sensation or leaky gut or migraines related to a DIO lack of activity. But I think that gastroenterologists do not do systematic research on the activity of DAO. And I'm also interested in the mast cells activity syndrome. And here in France, I don't know if we have the same statistics, but I would like to know how many of my colleagues do measure the DIO activity in order to measure as well dysbiosis. I think this is something significant for tomorrow. And I also think that we need to be able to diagnose the deficiency of the activity of DIO. I am also a professor in many different colleges, and I would like to insist very much in the study of the DAO activity and also the SIBO, the small intestine growth, bacterial growth. Also, medication. Medication can also be a cause of the inactivation of DAO. The echogenetics, I think that there's a presentation later on the relationship with the genes. And also alcohol is one of the main causes of DAO inactivation. So before prescribing a supplement of DAO, we have to eliminate the factors that lead to the inactivation of DAO. And we have to bear in mind as well the ethiopathological factors for the reduction of DAO activity. And thanks to our Spanish colleagues that organize this Congress, we now have more solutions. Before, we could only treat dysbiosis as we did. And actually, I would like to present at the end of the presentation a clinical case. And it's a case of a patient with whom that I treated, and we could not really treat those patients well before getting more research on DAO. So I think that systematically we need to try to find the cause of DAO inactivation before rushing to give supplements. So again, I was wondering whether here we're talking about bacteria inhibiting HNMT, and why do I have this DAO deficiency? Is this induced by the presence of certain bacteria, aerobic and anaerobic bacteria? So thank you very much, Dr. Ra, for really opening this gate. I've taken very good note of what you were saying, and actually this activity of DAO is very much related to this presence of aerobic and anaerobic bacteria. So this is a field that is really very interesting to try to find this balance in the microbiota. And now in the pathway of methylation, here we're talking about an enzyme which is 90% hepatic. So it's actually because of histidine decarboxylase that we will try to change the activity of DAO. I am not going to dwell on what Sonia Sanchez presented. It was really fascinating. And here we can see the reduction of Prevotella, Ruminococcus, and Faeculobacterium. And we have here this strain of bacteria. And I've also seen a research lab near Paris, and I think that they've done pretty well because they have isolated this bacteria and At that time, nobody believed in this, but actually these bacteria are today mentioned as responsible for many different diseases. And I can find them systematically in the microbiota. And Prevotella is also present in African populations, and I can see as well that vegetarians and vegans, they have a higher Prevotella than omnivorous and carnivore people. And I'm not saying here that we do not need to eat meat. Anyway, so there's this abundance of pathogenous bacteria that will generate alterations in the mucus, and it's going to generate as well alterations in Staphylococcus and Proteus, and this is what you were showing before. And I thank you for showing this excellent work here, the importance of protobacteria in the histamine intolerant group. And this is something that I look for systematically. But at the beginning, I was not so much aware of this. And today, I'm so happy to see that actually, well, this is a reality. This is being confirmed. It's the importance of this protobacteria in histamine intolerance. And I think that, well, Dr. Sanchez analyzed 50 different people. And there's this research group as well in Marseille, which is doing some research about this. And there's this lab, Alfabio, which does a lot of research and diversity of microbiota and the irritable bowel syndrome. So this is something that really has exploded. And today we're proposing 50, 60 bacteria that are responsible, but I'm sure that there are many other bacteria that we have not yet identified, or there's not enough study on this. But actually, anyway, this field of research is really evolving, and we can see as well that its fecal bacterium prosnitzi is very much related to this histamine intolerance. And here we've got another study that you already cited by Dr. Schenk with similar results, with this increased levels of protobacteria and with the action of zonulin. This is something that I found quite striking, and I'm wondering if there's a relationship here in the sensitive to histamine group and this increase of zonulin. I've also regrouped many different studies, and this is some kind of database on the research of different bacteria. And we see the same families everywhere. So the question that I have for Sonia later on is whether this typology of microbiota, which is for the histamine intolerance and that stimulates histidine decarboxylase. So is it really leading to an activation of histidine decarboxylase because of the presence of certain bacteria? So from eubiosis to dysbiosis. So, well, I thought that it would be worth explaining what dysbiosis is, because if we try to understand how dysbiosis appears, maybe we can try to treat it. There are some heavy treatments. There's also fecal transplantation. Well, dysbiosis is a loss of biodiversity of the microbiota because of endogenous or exogenous facts. And this is going to disrupt the balance of the microbiota, or it's going to lead to a loss of diversity. So this will lead to a lack of regulation of the health system. We see that the microbiota is always different depending on the way we eat. We know that hunter-gatherers had much more diversity of microbiota. Today we've lost 50% of that diversity. There's also the appearance of antibiotics in foods and in people, actually. I may remind you that the range of intervention for microbiota is from 0 to 2 years. After 2 years, this microbiota is very well established. And there can be some transient microbiota, but our fecal signature is built by our mother when we're born. And this is going to be developed up to 2 years of age. And after that, it's very difficult to change that. So we need to protect and preserve this microbiota. And we also know that those children who are born after a C-section, they are at a higher risk of developing histamine intolerances and other pathologies. So it's very important as well during the anamnesis to know how that person was born. I mean, how the delivery was performed. And also there's the influence of diet on the microbiota, and we know that the Mediterranean diet is the most beneficial one. The Western diet is very deleterious for our microbiota. This has been studied very well already, but it's very important to insist on this. And we see that in all cases of dysbiosis, there's an increase of proteobacteria. And so we find fecal signatures which are quite similar in the cases of histamine intolerances, in the cases of irritable bowel syndromes. We find this kind of dysbiosis, this pro-inflammatory dysbiosis, and this is going to disrupt all the immunity system. We also know that dysbiosis has effects in the mucus layer, in the motility, in the digestive motility, and there's also painful symptomatology. So we understand that histamine intolerance, the intolerance to certain foods, and the celiac disease, all this has some consequences, and they're not only biological. And actually, I think that if we are able to regulate the dysbiosis and go back to a balance, it's all for the better. There's also other causes, such as taking medication, allergies, the presence of pesticides, the lack of fiber in our Western diet, maybe an excess in physical activity or chronic infections. I know that 25% of irritable bowel syndromes are triggered by gastroenteritis that have not been treated correctly. And this could be an open gate to this histamine intolerance as well. And there can also be some intoxications, acute intoxications to salmonella, for instance, and this could induce histamine intolerance. Also, stress is at the heart of dysbiosis. And there can be techniques to reduce the stress, regardless what the pathology is. There are many different services and wards, especially at the University of Grenoble, who are working on the vagus nervous system in order to improve the situation of stress. So there are some clues to follow, really. And regarding this intestinal dysbiosis and the causes, there's a lack of chewing when eating. There's also a deficiency of enzymes at a pancreatic level, and also the acids, bile, for instance, and we know that this is very much related to the microbiota. And if there's a dysbiosis, you do not have this biliary acids that you can regulate. So there's all these open gates that create this histamine intolerance. There's also emotional factors such as stress. There's hypochloridria as well, which can be induced by the foods, but there's also this proton pumps, which can lead to this hypochloridria, to this lack of hypochloridric gastric, and this dysbiosis and the situation of putrefaction that is very difficult to treat. And we really have difficulties to correct this kind of problems, such as hypochloridria and poorly healed gastroenteritis. Also, well, the consequences of dysbiosis. We have, for instance, histamine intolerance, but it's not only this. We also have to bear in mind overweight, metabolic syndrome, stress, anxiety, depression, small bowel dysbiosis, such as SIBO, which is a pathology that is not so well known, a little bit more nowadays in France by gastroenterologists. And we try to assess this small bowel dysbiosis. This small bowel dysbiosis, whether it is bacterial or not, well, this can induce histamine intolerance. And this is actually something about which I wanted to ask Dr. Sanchez. I'm actually trying to speed up, not to exceed the time that was given to me. So the symptoms of dysbiosis, you've already talked about this, but now here I would like to tell you how to differentiate these two kinds of dysbiosis. There's the fermentation dysbiosis and putrefaction dysbiosis. And in our case, regarding DAO deficiency, we have tried to see whether it's we're talking about fermentation of putrefaction dysbiosis, or maybe a mix of those, because we're talking about these protobacteria that are increased, and we know that there are proteinogen foods and histamine-rich foods, and maybe we're dealing here with dysbiosis, putrefaction dysbiosis, rather, when it comes to histamine intolerance. So too much proteins, belly gases, and that come from this putrefaction. So regarding IBS microbiota and histamine intolerance microbiota, well, in my research group with a Belgian lab here in Brussels, we were interested in the fecal signature of the microbiota in the case of IBS. And we saw that it was quite similar. We have this bacteria having a negative health effect. We have this increase in this kind of bacteria. And maybe this is not exactly what leads to signature of the microbiota in the case of IBS. However, what is similar is that we're always going to find a decrease of Faecalibacterium proudnitzii, and this is similar for histamine intolerant people. And this is not always the case for IBS. And for Akkermansia, well, we have a team who was working on this strain, Akkermansia. And well, we know that we need to have the right balance of Akkermansia. In Western populations, we're deficient in Akkermansia. And we have shown a correlation between reduction of Akkermansia and an increase in metabolic syndrome. I can tell you that it's a very complex field of expertise, because it's difficult to know which are the bacteria which are deficient or which are excessive. So we will see that the therapeutical approach is very complex, actually. We see that many people are treating small intestine overgrowth with antibiotics, and I'm really wondering whether this is the right way to do. So are we going to give prebiotics, probiotics, postbiotics? And there's also this issue, the ecocerticum. I think that this is a very interesting way to treat this kind of patients. Also, well, the microbiota profile in histamine intolerance, we've been talking about this, Mrs. Sanchez has told about this, and but we need more studies with a larger number of N number, but we can already see some typologies of microbiota for irritable bowel syndrome, for example, and now for histamine intolerance. And I really thank you very much for this work, because it has really shed some light about how I treat dysbiosis and how I try to reach a balance again. But I can tell you that it's not easy. It's not easy to also to grow certain bacteria which grow in an anaerobic medium. And as any other plant, they need to be fertilized. And I have found some prebiotic fibers which are very interesting, or some resistant starch which are interesting in order to feed this kind of bacteria, in order to try to awake this flora that is a little bit lazy. So we can bring some substrates that can be interesting. We can try to imagine a prebiotic that will lead to better results for histamine intolerance. We have been talking about ruminococcus as well, which is often reduced, and there are other beneficial bacteria. And certain bacteria, we know how to increase them, but Professor Renaud, you need to wrap up. Thank you very much. Because we have exceeded our time. So for Prof. I'm going to try to summarize here. We've got the microbiota signature in case of irritable colon. And for the mechanisms of action, well, I'm not going to reveal this now because I have no time, but here I'm going to show you the microbiota in the case of IBS, and here we've got this effect in proteobacteria. So today we have very interesting paths for research about the relationship between dysbiosis and histamine intolerance, and the activity of decarboxylase as well, histidine decarboxylase. And what about our lifestyle? What about what we eat and the relationship with irritable bowel syndrome, histamine intolerance, and other pathologies? Maybe if we could try to think about other functions, such as enzymatic function, these dysbiosis could be reduced. And we could really develop solutions for the leaky gut. So, well, this is the end of my talk. Thank you very much. I hope I didn't take too much time. Actually, well, the product that I wanted to tell you about is this product, which is interesting. So, well, thank you very much for your attention. I think that's all for me.